All right. So today's 10th journal club is on the topic effect of plant-based diets on gut microbiota, which is systematic review. Today's journal club study presentation is going to be done by the following participants as follows. The study introduction will be done by Dr. Suleiman, and the methodology will be done by Dr. Nitish. Results will be done by Dr. Melinda and Dr. Sandhya. Discussion will be done by Dr. Sindhura, it's me, and conclusion will be done by Deshna. So this is the flow of the presentation in today's session. So Dr. Suleiman, could you please take it over, please?
Good evening, everyone. I'll initially do the small brief introduction for the PAN team, followed by the introduction for today's Journal Club. Uh, Greetings, everyone, in the 10th Journal Club of PAN India, and I am Suleiman, a second year student of Bangalore Medical College and Research Institute, and a part of a team that's working towards better daily dietary choices. Since prevention is always a better option than cure, it forms the fundamental idea of PAN organization. It's quite a spirited team working towards an approach to build a network globally and nationally to improve the nutrition system with the power of right dietary choices and to prevent diseases. Since always a change is contradicted, hence the PAN team works rigorously to appeal to society through evidence-based guidance. And with the same very purpose to reach out nationally and spread this positive movement, PAN India was established in September of 2022 with chronic diseases, cardiovascular diseases, diabetes reaching new heights. Pan India has got an aspirational approach organizing polls, quizzes, feedback forms, query answer sessions, journal clubs, in person CMEs to disseminate this information to a wider group through various means of social media and, as I mentioned, in person CMEs too. The PAN team is headed by our director, Dr. Rajina Shahin, and it's also got a lot of spirited volunteers, interns, ambassadors, which include professionals right from medical students in yet in med school to working doctors, nutritionists, and all much with the goal of improving the broken nutrition plate. And moreover, it especially includes all of you who attend the PAN events and form the backbone of methods to spread our message. And since it's quite evident that the team is showing a positive growth and we are having more and more people joining in. So anyone who's interested with the same goal, you're always welcome to join and be a part of this movement. With this brief introduction of the PAN team, now I will be moving on to today's journal club, which is on the topic of uh, im- plant-based diets and the effect on the gut microbiota. Next slide, huh? Okay. So beginning initially, I'll give a brief introduction of what are the major prominent enterotypes in the human microbiome. It includes the bacterioids, Ruminococcus, and Prevotella. And it's been seen that a diet that is highly rich in animal protein increases a few bacteria, but then the bacteria that it increases in a way have a negative impact on health because animal proteins also decreases the beneficial bacteria, which include lactobacillus, progeria, affecting the overall diversity in the gut microbiome. To get this again in the evidence perspective, a study was conducted on mice who were fed with a high fat, high sugar diet, and there was a reduction in the prominent human microbiome with a beneficial. Next slide. To get a better emphasis of this, let's compare a Western diet which is prominent with processed food, prominent with more of animal-based proteins, and comparing it with plant-based diets, which is more prominent in purely plant source foods, vegetables, and fruits. So it was noticed in the gut microbiome that there's increase in opportunistic bacteria and bacterial metabolites in Western diet, which is prominent with non-plant based sources, an increase in lipopolysaccharides, inflammatory cytokines, and an overall decrease in beneficial bacteria. All of these, which cause an increase in opportunistic bacteria, inflammatory cytokines, and a reduction in beneficial bacteria, cause an increased risk of nutrition-related chronic diseases, which very much include the common ones which are rising every day, like diabetes. Comparing it with plant-based diets, there's a reduction in opportunistic bacteria which are harmful, reduction in lipopolysaccharides, and reduction in inflammatory cytokines, whereas there's an increase, a positive way, in beneficial bacteria. Overall, this causes a decreased risk of nutrition-related chronic diseases. As coming to what this study was, there was a study done in Burkina Faso and Italy. Burkina Faso is an African nation, and Italy is more of a Western, where they follow more of animal-based diets. So, European children consumed a Western diet which is low in fiber, while the Burkina Faso were rich. The children were rich in fiber and resistant starch. Researchers, when they studied both of these, found that the Burkina Faso children had a microbiome enriched with Bifidobacterium, Prevotella, and Xylanobacter, while they were depleted of firmicutes. So overall, this indicates that the natural microbiome is being promoted in a plant-based diet. Next, please. So as we come to today's study, which is titled as Effect of Plant-Based Diets on Gut Microbiota, a systematic review of interventional studies. This was done in the Jeffrey Shai School of Medicine and Health Sciences in Malaysia. It emphasizes on the relationship between plant-based food items and the gut microbiome, along with including the biochemical and anthropometric measurements as secondary findings. To give a basic of the available plant-based diets, because it's not necessarily that there's just a vegetarian and a non-vegetarian diet. It includes a broad mm, spectrum over here. Now we can see plant-based diets of vegetarian incorporate most of the plant sources, while they try to exclude the products of animal origin, which include red meat, fish, poultry, eggs, and even dairy. A flexitarian or semi-vegetarian diet. It includes a little bit of meat, dairy, poultry, or it can even include seafood. While pescatarians, they completely avoid meat and poultry, but then they do consume fish and seafood. Lacto-ovo-vegetarian, as it's quite evident from the name, they include eggs and dairy, but they exclude meat, fish, poultry, and seafood. As we speak about plant-based diet throughout our journal clubs, so it's plant-based diet. The ideology is gaining popularity, with almost one-fifth of the Asian population already embracing vegetarianism. And the COVID-19 pandemic, although it's got a lot of negative impact, it's also got a positive impact that even the American households, the Westerns, have purchased plant-based foods, including milk, and which have an alternative for meat too. Overall, not just health benefits, plant-based diets have got environmental benefits too, as they decrease the carbon emissions produced mainly through rearing animals and livestock. And they are also lower in saturated fats and high in fiber and phytochemicals, which are beneficial for the human health. Now I've done the introduction for today's brief introduction for today's studies. Materials and methods coming was a systematic review and meta-analysis. And from this paper, the results, the discussion, and conclusion overall will give a better idea about how switching to a plant-based diet will help increase the diversity of health-promoting bacteria in the gut. Over to the materials and methods. Thank you.
Thank you, Dr. Suleiman. Good evening, all. I'm Dr. Nitesh, and today I'm going to present methodology part of this systematic review. So, while conducting this systematic review, Prisma guidelines were used. This systematic review protocol had been registered with Prospero. Prospero is an international prospective register of systematic reviews. Registering this protocol with it means with a Prospero is not mandatory, but it is generally accepted as a good practice and which helps to produce unbiased piece of work. Now coming to the articles for this systematic review, articles were retrieved from databases like Ovid Medline, Scopus, PubMed, and Embase. And they also retrieved articles manually using references. In search strategy, they used mesh terms and keywords such as plant-based, vegetarian, vegan, gut microbiomes, prebiotics and probiotics, and intervention. Next slide, ma'am. Okay. Now coming to study selection, it was done using Covidus platform, and during this process, duplicates were removed. Two authors independently evaluated for titles and abstracts, and also evaluated full text criteria. And the third author resolved any conflicts encountered during both stages. Next slide. Now eligibility criteria. As per the study design criterion, intervention studies including randomized control trials, non-randomized trials, and pre and post intervention studies included in this review, whereas non-peer-reviewed publications were excluded. Studies where study population was adults on plant-based diets or who were counseled to eat plant-based diets were included, while studies where study population was on single type of food were not considered for this review. As per the outcome criterion, studies where effects on gut microbiota recorded, and this was done using metagenomic sequencing analysis were included in the study, while studies which did not recorded effects on gut microbiota were excluded. Next slide. Uh, the data extraction and synthesis was done by authors independently. For this systematic review, extracted data included author, country, study design, number of participants, primary and secondary outcomes. For primary outcomes, they considered gut microbiota changes, including relative bacterial abundance and diversity levels, whereas for secondary outcomes, they consider changes in metabolic parameters and changes in weight. The qualitative synthesis of data on type of gut microbiota and their correlation with outcomes were done. Next slide. Finally, coming to quality assessment and risk of bias, it was done by two reviewers independently. For the quality assessment of randomized control trials and pre and post studies, they used NIH quality assessment tool, while for the quality assessment of non-randomized trials, they used Robbins I tools, that is risk of bias in non-randomized studies of interventions. The quality assessed was based on study population, eligibility criteria, sample size distribution, time frame, exposure, outcome details, and other sources of bias. Based on quality assessment, the studies were categorized as good, fair, and poor. So this was the methodology they used to conduct this systematic review. Now I request my colleagues to continue with results.
Thank you, Dr. Nitish. I would start with results now. Now coming to the study selection process, the database search found 203 records, out of which 102 were duplicate entries and 101 titles and abstracts were evaluated. Out of this, 23 full texts were reviewed using their review eligibility criteria. A manual search yielded 6 additional articles. One was excluded after not fulfilling the criteria. Now, most reports were excluded because either there was no plant-based intervention or did not find the reportings on gut microbiota or were conference experts. Now, finally, only 12 studies matched the inclusion criteria and was included in the systematic review. Coming to the study characteristics. All the 12 included studies were assigned to interventional study design. 2 studies were published in 1990s, 3 were published between 2010 to 2018, and all others were derived from the year 2020 to 2021. The geographical origins of the included studies were very diverse. Like 10 studies were conducted in Europe, particularly in the United Kingdom, Sweden, Finland, Germany, and Italy. 4 were from United States of America, and 1 was conducted in Asia, that is particularly in Korea. Now over here you can see the sample sizes range from 6 to 168 with total of 583 participants and comprise of healthy, obese, cardiovascular risk and rheumatoid arthritis individuals. All studies reported the inclusion of men and women except for one which did not report anything on sex. The ages of the participants ranged between 21 and 61 years. The duration of intervention varied from 5 days to up to 13 months. 6 studies reported vegan dietary intervention. 3 studies investigated the effect of ovo-lacto-vegetarian dietary interventions and plant-based diets on gut microbiota composition. 6 studies performed 16S RNA sequencing to detect the gut microbiota composition, except for 2 studies that use gas liquid chromatography to investigate the role of microbial metabolites. 4 studies use both the methods, that is RNA and gas liquid chromatography. Coming to study quality assessment. Mean score on NIH quality assessment scale for intervention studies was 58.2%. 4 studies had good quality and equally 4 were acceptable and 1 study was poor. Mean score of pre and post study was 50%. Next slide, please. Yeah. Now coming to the gut microbiota composition in vegetarian and vegan diets. Yeah. So this is the pyramid diagram you can see over here, which shows the bacterial taxa in vegetarians and vegans, which is compared to omnivorous. Now this green color which you can see is because of the higher abundance of these bacterial taxa in vegetarians. Now if you see, it is divided among class, family, genus and species. Now to give you a few examples, like in class, the major ones are Clostridia and Bacteroidea. In family, the microbacteria are mostly Lachnospiraceae, Enterobacteriaceae, Ruminococcaceae. If you look at the genus, the most common ones are Roseburia, Romanococcus, Streptococcus ones, which we already know about. And coming to the species, you can see Lactobacillus, Faecalibacterium prausnitzii, and Bacteroid speculus. So this shows higher abundance because of the green color in this slide. Ma'am, next slide. Now over here, you can see a same pyramidal diagram, but it is showing red color, which the red color indicates it is lower abundance of this bacterial taxa in the vegetarian and vegan compared to the omnivorous. Now over here also, like a few examples, in class you can see Bacteroideae, in family you can see Enterobacteriaceae, Bacteroideaceae, Acidomyinococcaceae. In genus group you can see Faecalibacterium, Parabacteroids, Bifidobacterium. In species you can see Elistipes, Parabacteroids, Coprococcus, Streptococcus. So these are few examples which are lower in abundance in vegetarians compared to the omnivorous. Ma'am, next slide. Yeah. Now at individual level, we'll see how it affects. Now we had seen that Enterobacteriaceae is more prominent or is higher in abundance in vegetarians and vegans. But there was one study which showed contradictory results, which showed that there was higher abundance of Enterobacteriaceae in adult with cardiovascular disease risk with a plant-based diet, and also there was low abundance in vegans. Only one study showed that, but rest all others proved that Enterobacteriaceae is abundant in vegans and vegetarians. Now, another 4 studies, they found statistically significant increased levels of Ruminococcaceae, whereas at the same time, reduced levels of Bacteroideaceae in vegan and plant-based diets. Ma'am, next slide. Yeah. Now, this is one more study conducted by Ahrens et al., which was done in individuals who followed a 6-day plant-based diet. Now, in this, you can see there was it showed a certain increase in certain type of gut microbiota and reduction in some. Now, the increased ones were Lachnospiraceae, Ruminococcaceae, Monoglobaceae, Egerthalaceae, Cristansenalaceae, Butyriceae coccaceae, and Erysipelatoclostridiaceae, whereas it showed reduction in Barnesilaceae, Pseudorlaceae, Marinifilaceae, Tenorlaceae, Ricinolaceae, and Acidomyinococcaceae. Ma'am, next slide. Yeah. Now, this is based on the genus criteria, the genus category of the gut microbiota. Now, you can see vegan diet, plant-based diet, and animal-based diet. Now, Faecalibacterium, which is the common cell, it is shown that it is decreasing in vegan diet but increasing in plant-based diet. Whereas Elistipes, it is increasing in vegan as well as animal-based diet. But according to the Ahrens et al., it shows that in plant-based diet, there is reduction in this particular bacterial group. Then bacteroids showed increase in vegan and animal-based diet, but reduction in the plant-based diet. Coprococcus showed increase in vegan, but decrease in plant-based diet. Ruminococcus and Roseburia, both of them, they showed increase in the plant-based diet and also in ovo-lacto vegetarian diet. Ruminococcus also showed increase in vegan diet, whereas roseburia showed decrease in vegan as well as in animal-based diets. Next slide, please, ma'am. Yeah. Now coming to the species. Now, Faecalibacterium prosnisi species was significantly increasing plant-based and vegan diets. Now, this particular species is one of the most prominent in your gut microbiota. So what happens is this protects your mucosal layer of your gut. So if this bacteria decreases or gut microbiota decreases, there are high chances of leading to inflammatory bowel diseases because it is an pro-inflammatory bacteria. Now, Bacteroides fragilis was increased in vegans, but according to Keliova, it reported a significant reduction among the low-fat vegan diet group. Next slide. Yeah. Now, coming to the association of plant-based diet and gut microbiota on metabolic, inflammatory, and body composition in healthy and unhealthy individuals. Next slide. Yeah. Now, over here, you can see the health benefits given by the microbial metabolites. Like anti-inflammatory effect, immunomodulatory effect, systemic anti-obesogenic effect, anti-hypertensive effect, hypocholesterolemic effect, anti-proliferative and antioxidant effects. Now, in this diagram, you can see a few examples of gut microbiota which help in certain degrees, like which I have spoken about already was the first one, Faecalibacterium prosnisi. It inhibits the production of pro-inflammatory cytokines. It also leads to butyrate production. Secondly, you can see over here Escherichia coli, which leads to bacteriocin production, then also protects against pathogen colonization. Then next is Bifidobacterium longum, which shows improved intestinal barrier function and inhibits inflammation. Lastly, we have the bacteroidetes theta iota omicron, which leads to nutrient absorption, maturation, and maintenance of the epithelial cells. So these are a few examples. Now, as I've already mentioned, these effects depend on the composition and the substrate of the microbiota, and also on they are influenced by the diet. And the changes, they arise from the control of gene expression, metabolism, and intestinal function. Next slide, please. Yeah. Now, coming to this slide, you can see it is showing the qualities between healthy and unhealthy individuals and comparing it with three parameters. One is the microbiota composition, biochemical composition, and anthropometry. Now, if you see the microbiota composition in both healthy and unhealthy individuals, there is significant increase or there is no change in the diversity of the gut microbiota. But there is a significant decrease in alpha diversity in the unhealthy individuals. Comparatively, there is no change in alpha diversity in the healthy individuals. Also, there might be no change in the richness of the gut microbiota, whereas there will be increase in richness of the gut microbiota in the unhealthy individuals. Looking at the biochemical parameters, there is a significant decrease between unhealthy and healthy individuals. First, coming to the healthy ones, there is increase in the short chain fatty acids. There is increase in butyrate production pathway. There is also increase in acetyl-CoA and X4-aminobutyrate succinate pathways. It also lowers postprandial glycemic response. There is lowered inflammatory markers concentration. There is improved cardiometabolic health. Also, there is diversity of T cell repertoire and lower expression of IgE antibodies. Comparing it with the unhealthy individuals, you can see there is decrease in the acetate and butyrate and propionate. There is no increase in the short chain fatty acids. There is no change in the plasma TMOs. There is also, or in the acyl carnitine or choline, there is increased level of plasma 1-carnitine, improved cardiometabolic health similar to that of the healthy ones, but there is lowered inflammatory markers concentration. Lastly, coming to the anthropometric parameter, there is either decreased or no change in the BMI in healthy individuals. But whereas in unhealthy, there is decrease in the LDL cholesterol BMI. There is also decrease in systolic and diastolic blood pressure. With this, I would like Dr. Sandhya to take over. Thank you.
Thank you. I'm continuing with the results, but am I audible?
Yes, you're audible.
Okay. Yes. So, continuing with the results section, I'm talking about the effects of plant-based diet on the gut microbiota and the metabolism of our body. So there is a study done by David et al., which have showed the following results: that on consumption of the plant-based diet within a short period of time, our gut microbiota rapidly adopted to that change, and the changes were so rapid that they were visible within 5 days of the consumption of the new diet. Then, on consumption of the group which have consumed the animal-based diet, they have shown a more significant effect on modifying the gut microbiota. They have seen that of the vegetarian group, in one vegetarian individual in the study was switched to an animal-based diet had shown the most significant decrease in Prevotella. Coming to the next slide. Yes. Now there are a group of studies which have observed positive effects of the vegan diet on the metabolism of our body and microbiota. They were the studies done by Kim et al., Paglia et al., and Kelly et al. So their results are as follows. Next slide. Yes. The study done by Kim et al., they have taken 6 individuals who were obese or diabetic with or without hypertension, and they made them to take vegan diet for a period of 1 month. At the end of 1 month, they have seen that in those 6 individuals who have consumed, there were lower blood glucose levels, lower body weight, low triglyceride levels, and reduction in total cholesterol, reduction in low density cholesterol, and also reduction in HbA1c was observed. And they have seen that in the gut microbiota, they have seen that the Enterobacteriaceae family of bacteria, which is known to cause the chronic inflammation in the body, was found to be reduced in those who are following this vegan diet. Next slide. Next, in the study done by Paglia et al., they have also taken 2 groups, one who is taking the Mediterranean diet, which include the meat, and other taking the vegan diet. And they have seen that after doing a correlation analysis, there was a positive association in the changing on the bacterial taxa and variation in the clinical and biochemical indicators in a positive way. That is same thing like in the Kim et al. Study. They have also seen that there was reduction in the basal metabolic index. They have seen there is a reduction in the body weight. There was a reduction in the HbA1c value, all these things. But they have not found any significant difference in SCFA production, the short chain fatty acid production between the Mediterranean or the vegetarian group. Next slide. In the study done by Kelly et al., again, when they have taken these 2 groups, vegan and Mediterranean group, and they have observed the effects, they also found the similar results like the Kim et al. And Paglia et al. There was a reduction in the weight of the body. And also one important indicator they have observed is there was increase in insulin sensitivity. That is, there were decrease in, they have observed that there decrease in insulin resistance in the individuals. And coming to the study of effect of gut microbiota, they have seen that those who are consuming this vegan diet, they have seen that there was increase in Faecalibacterium. There was a relative abundance of Proteobacteria decreased after the 16-week vegan diet. And they have seen that in the group who are taking the meat, the butyrate producing bacteria, they were reduced drastically. Next, coming to the next slide. Is next, there were studies which have studied the effect of vegan diet on the cardiovascular disease risk. That one of those studies is Dijik et al., and they have seen that they have taken 2 groups: study subjects with ischemic heart disease who were kept on the vegetarian diet, and the study with subjects with ischemic heart disease who were kept on the diet that is non-veg diet or the diet which includes meat. And they have seen that after this intervention, the following effects were studied: that there were low levels of low density lipoprotein, there was low cholesterol and low body mass index in those ischemic heart patients who have consumed vegan diet. And they have seen the following effects on gut microbiota. They have seen that their subjects on the vegetarian diet had a lower relative abundance of fecal microbial taxa and the compounds which are linked with metabolic disease, particularly the cardiovascular disease, than those who are on a meat diet. Next slide. Similarly, there is another study, Herons et al., which my colleague was also discussing in the previous slides. They also studied the effect of the vegan diet in patients with CVD risk. And when they have put the patients with CVD risk on plant-based diet and exercise, both combination, so they have seen the following effects. There was statistical significant decrease, the p-value being less than 0.05 in blood pressure and lipid profile too, and reduction in total cholesterol and triglycerides. And one important effect they have seen on gut microbiota is there was significant increase in butyrate producers, namely the lactose and oxalococcyles. Yes. Finally, so they have come to a conclusion that the change in the microbiota was substantially linked to this. Because of taking the vegan diet, there was a change in the gut microbiome composition, and this has been linked to all these changes in the metabolism, that is the decrease in BMI, the blood pressure, the lowering the cholesterol and triglycerides. Next slide. But there were two studies. All these studies have shown a positive association between the vegan diet and lowering the BMI and all. But there were two studies in this research. They have not found any significant decrease in any BMI or weight following a vegan diet. But the Zhang et al., they have observed one thing: even though there is no significant difference in BMI or weight, they have seen that there was less diverse T cell repertoire, and they have reduced the levels of IgE, crucial allergy-related immunobiological modulator. Next, there were studies which have shown the association between the plant-based diet and their effect on the inflammatory process in our body. They were Peltonen et al. And other studies. In Peltonen et al., what they have done is they have taken 53 rheumatoid arthritis patients, and what the intervention they have done, they have transition of these patients. They have put these patients on a vegan diet for a period of 1 year, and at the end of the 1 year, they have seen that there was a significant difference in intestinal microbiota. Similarly, like Peltonen, they have seen other studies which also put the rheumatoid arthritis patients on vegan diet. They also shown the same thing. Even within a short period of time, like within 1 month of the vegan diet, it was enough to significantly change the microbiota and result in decrease in inflammatory markers and decreased inflammation in the body. So concluding all the above studies, they have come to a conclusion that vegan diet has a role in bringing the rapid change in fecal microbiota in a short period of time. Thank you. Now my colleagues will continue with the discussion part.
Yeah, thank you so much, Dr. Sandhya. Good evening, everyone. I'm Dr. Sindra. I'll be doing the discussion part. So the current topic that we're covering is the relation between the dietary pattern and the gut microbiota. So first, let's understand what is gut microbiota. The gut microbiota is a complex community of bacteria which are residing in the intestine, and the relation between the gut microbiota and human health is increasingly recognized these days. And it's now well established that a healthy gut flora is largely responsible for our overall health of the human being. So though the gut microbiota in an infant, if we talk about the gut microbiota in an infant, they appear haphazard during the infancy, but they are going to resemble the adult flora by the age of 3 years. So if you look at the bacterial load or the gut bacterial load, in the stomach, it is significantly lower, and the load exponentially increases as we move down the tract, like from stomach to duodenum to jejunum to ileum, and ultimately in the colon, the bacterial load is going to be around 10.9 to 10 power 13 bacteria. And the normal gut microbiota, it imparts specific functions with respect to host nutrient metabolism, drug metabolism, the maintenance of structural integrity of the gut mucosal barrier, the immunomodulation, and the protection against the pathogens. So there are several factors that play a role in shaping the normal gut microbiota. So both human and animal studies have demonstrated that diet can influence the composition and functioning of the gut microbiome. Other than the diet, the other factors that can that can influence the composition and the functioning of the gut microbiome are the genetics, the mode of the delivery at birth, whether it's a normal vaginal delivery or a C-section, and the method of infant feeding, whether the baby is breastfed or formula fed, and the use of medication, specifically antibiotics. So these are all the factors that can contribute to the composition and the functioning of the gut microbiome. As we know, diet is one of the main driving factors contributing to the composition and diversity of the gut microbiota. In response to the diet, the gut microbiota also starts producing various metabolites. So if we take a diet rich in complex indigestible carbohydrates like dietary fibers and resistant starch, they support the gut microbiome derived metabolites like short chain fatty acids. Among that particular group, the notable ones are acetate, propionate, and butyrate. And among those three, the most notable one is butyrate. So conversely, if you look at the diets that are low in fiber and high in fat, and which are composed of simple carbs, they have a low short chain fatty acid fermenting capacity, and they are linked with chronic health conditions such as colorectal cancer and cardiometabolic diseases. So this current systematic review, it is the first one to assess the association between vegan and vegetarian diets on gut microbiota composition and human health outcomes from just the intervention studies. So the results suggest the presence of more bacteria that can break down the fiber in vegan or vegetarian compared to the animal-based diets. So if you look at the vegan and vegetarian diets, they are rich in dietary fiber fermentation products and other carbohydrates that can produce SCFAs or short chain fatty acids. Now, thus in individuals who adapt a plant-based diet, an associated rise in SCFAs can be seen, and it also showed a negative correlation with pro-inflammatory cytokines that can lead to inflammation as well. So on the other hand, when someone is following an animal-based diet, it resulted in a significantly lower levels of carb fermentation products like SCFAs and all. So which can also lead to increase in the inflammation. Now here in this beautiful image, we can see when someone follows a diet rich in fiber, we can see the positive kind of a good gut bacteria developing, which are going to lead to the derivation of the food metabolites like short chain fatty acids, predominantly acetate, propionate, and butyrate, which can lead to the anti-inflammatory, anti-anxiety, and all the various properties that have been discussed already by my colleagues. And when someone consumes red meat and high fat diet, so it can lead to the opposite, where it increases the production of the metabolites like TMAO, L-carnitine, choline, and all, which can, or cytokines, lipopolysaccharides, or long chain fatty acids, which can actually increase the inflammation, thrombosis, and a risk of atherosclerosis. So here also, when we consume a food that is rich in dietary fiber, it is going to impact the diversity and the richness of the gut, the composition, the richness of the gut microbiota, and that leads to a better fermentation and the production of the short chain fatty acids, which again can reduce the colonic pH, as well as it also portrays the anti-carcinogenic effects as well. Not only that, when someone consumes a food that is predominant in dietary fiber, as I've already mentioned, it's going to increase the fecal bacterial diversity, the bowel movement increases, the short chain fatty acid production increases, the good gut bacteria increases, the body weight reduces, the inflammatory cytokines are going to reduce, the bloating is going to reduce, the secondary bile acids, which can lead to the colon cancer formation, they can also be reduced. So among the SCFAs, the most abundant one, as I've mentioned, are acetate, butyrate, and propionate, which together constitute approximately 90% of the SCFAs produced by the microbiome. And these SCFAs, they promote human health in a variety of ways by providing energy for the intestinal cells or the colonocytes, and they maintain the gut barrier, and they also activate the anti-inflammatory signaling cascades. The low levels of SCFA producing microbiomes have been associated with diseases such as type 2 diabetes, inflammatory bowel disease like ulcerative colitis, Crohn's, asthma, Crohn's and asthma separately, etc. And in addition to the major short chain fatty acids mentioned above, there are also less abundant short chain fatty acids such as the branched chain ones, which are isobutyrate, isovalerate, and 2-methylbutyrate, which are produced from the fermentation of amino acids like branched chain amino acids rather than the carbs. But little is known about their effect on the human health. So this is another beautiful picture which summarizes everything. So when an individual consumes the food that is rich in dietary fibers, it can lead to the fermentation and the production of good gut bacteria, which can lead to the production of short chain fatty acids, acetate, propionate, and butyrate. All these three of them through the G protein coupled channels, these are the effects that they're going to produce on various systems. If you look at the liver, it's going to reduce the lipid accumulation, decrease lipogenesis, and with respect to pancreas, the insulin secretion and the beta cell function can be improved. With respect to adipose tissues, the leptin release increases, which can lead to better satiety, and the energy expenditure increases, the caloric expenditure increases, that promotes the weight loss, and the inflammation reduces, the lipid accumulation reduces, even the mitochondrial functioning improves, the insulin sensitivity improves, and the gluconeogenesis also improves. So not only that, the substantial evidence shows that the short chain fatty acids have a preventive effect against various diseases, including type 2 diabetes, inflammatory bowel disease, various immunological diseases. They're going to boost the immunity in the host. Not only that, they're crucial for the neuron functioning and the maturation and for maintaining the blood-brain barrier function. And they also promote the metabolism and play a significant role in preventing or treating obesity. So the considerable body weight reduction has been observed in vegan and vegetarian intervention diets, strongly support this particular claim. Now, among the short chain fatty acids, if we specifically talk about the butyrate, which is the main end product of microbial fermentation of dietary fibers in the human intestine, it's going to play an important role in the maintenance of intestinal homeostasis and the overall health status. So, so here we can again see the butyrate production. It can lead to, it is involved in the epigenetic regulation of various effects like anti-carcinogenic, chemopreventive effects, anti-inflammatory. It reduces the insulin resistance, the effect on, it's going to reduce the risk of cardiovascular disease, immunological disorders, and it has a neuroprotective effects. And the intervention studies included in this particular review have reported an increase in the butyrate producing bacteria like Ruminococcus, Lactobacillus, Roseburia, Blottia, Elistipis, Faecalibacterium, etc. So butyrate has multiple physiological activities, specifically providing the energy to the colonocytes, improving the intestinal barrier via the upregulation of tight junctions, as well as by lowering the systemic inflammation by preventing lipopolysaccharides from crossing the intestinal wall and entering the bloodstream. If you also look at the effect of butyrate on various systems of our body, with respect to heart, it's going to increase the cardiac function, reduce the uric acid, decrease the uric acid load, increase the antioxidant defense mechanisms, reduce the inflammation. When it comes to the blood vessels, it's going to reduce the angiotensin to improve the vasodilation, improve the nitric oxide production, improve the anti-angiogenic thing, as well as with respect to the atherosclerosis, it's going to reduce the cholesterol deposition, decrease the migration of macrophages, and decrease in the addition molecules. And as a result, it also reduces the risk of atherosclerosis. With respect to the liver, it's going to improve the insulin signaling, reduce the hyperglycemia. And with respect to the adipose tissues, it's going to, as mentioned, it's going to increase the leptin, the thermogenesis can be increased, the lipolysis can be increased. And with respect to the gut, most importantly, the gut permeability reduces, the endotoxemia reduces, the microbiome modulation increases, and the cholesterol uptake also reduces. So till now, we have been discussing predominantly on the gut microbiota and the short chain fatty acids. The other component that has been discussed in this particular systematic review, other than SCFAs, is TMAO. So the another possible beneficial linkage observed in these intervention studies was the correlation of TMAO measurement with the relative abundance of specific microbes. So the red meat consumption has been associated with elevated TMAO levels, which is going to further increase the risk of cardiovascular disease and the inflammatory bowel disease. So we can see here when someone consumes a diet rich in red meat, it's going to increase the TMA concentration, which is going to alter the gut microbiota, leading to again increase the production of TMAO, which further puts an individual at an increased risk of atherosclerosis, inflammation, heart failure, myocardial infarction, and stroke, all of those things. So this is also a beautiful picture, likewise what we have discussed in the previous slide. So when one consumes a food that is rich in the red meat and all, it's going to alter the gut microbiota, leading to the increase in TMAO, which is going to exacerbate the vascular inflammation, impair the blood vessels, disturbs the cholesterol homeostasis, and finally increasing the atherosclerosis, putting us at a risk of atherosclerotic cardiovascular diseases. And Dejek et al. Observed a significant correlation between the genus Bifidobacterium and several species of Lactobacillus and Rhinococcaceae. They also observed a reduction in the plasma L-carnitine, which is a metabolite found in the red meat, among the subjects with ischemic heart disease who have adhered to the vegan diet intervention. The study also suggests the metabolic benefits, like there is a reduction in LDL cholesterol, total cholesterol, and body weight in vegetarian diet interventions, supporting the beneficial role of gut microbiota in modulating the biochemical parameters associated with CVD risk. So here in this image, we can also appreciate by including the healthy plant-based diet, we will be able to inhibit the production of this TMA and TMA. So again, just the summary here, the plant protein increase SCFAs, which is going to reduce inflammation, improves the gut health. When we consume animal-based protein or and predominantly animal-based diets, it's going to increase the TMAO, reduce the SCFAs. When the TMAO increases, it increases our cardiovascular risk, whereas when the SCFAs reduce, it's going to increase our inflammatory bowel disease risk and the colon cancer. So the presence of SCFA, short-chain fatty acids, and TMAO metabolites in the plasma of vegan and vegetarian diet interventions suggest gut microbiota's role as a modulator of the diet-host interaction. Despite the identified all the above discussed mechanisms, still the interaction of gut microbiota with other metabolites like plant-derived polyphenols, isothiocyanates, vitamins, and intestinal aryl hydrocarbon receptors ligands is still lacking, and that is where we need much more interventional studies to evaluate the better possible linkage between the gut and the diet and the microbiota composition. So this completes the discussion part. Now Deshna will be taking over the limitations and the conclusion. Thank you.
Hello, thank you so much, ma'am. Greetings, everyone. Now I'm going to discuss about the limitations of this particular study. Starting with the limitation, most studies included in this particular systematic review had a low number of participants, which might have insufficient power to the study results. Besides that, only one study included participants from the Asian continent, thus calling into question the results of generalizability to the Asian population, owing to different dietary intake, environmental exposure, or lifestyle habits. Other than that, lastly, the third limitation of the study was antibiotic and other drugs are known to change microbiota makeup. Except for 3 studies, none of the other studies analyzed reported medication intake, which may have contributed to the disparities in the outcome of this analysis. Next slide, please. Now I'll start with the conclusion of this particular systematic review and meta-analysis. Nutrition is an important determinant in the makeup of nutrients into active postbiotics for humans. Current research suggests that switching to a plant-based diet may help increase the diversity of health-promoting bacteria in the gut. However, more research is needed to describe the connection between nutrition, the microbiome, and health outcomes because of their complexity and individual heterogeneity. Next slide, please. Thank you so much. Now I'll talk about the takeaways for this particular the overall JC. Takeaway, next slide. What we see here is on the left side is Healthy Eating Plate by Howard, which suggests that half of the plate should be vegetables and fruits, which is ideally a good source of fiber. Along with that, 1/4 of the plate should be whole grains, and the other remaining portion should be healthy protein. On the other side, it is by NIN ICMR. That is, NIN is National Institute of Nutrition by ICMR for India. It is, when we look at the plate, it is similar to the Harvard plate in the sense that 50% of the plate should include fruits and vegetables. Here they have specified regarding intake of whole grains being around 25% and inclusion of healthy fats being 27 grams for the whole day. and a small amount of pulses and the source of protein and nuts and oilseeds. Here they have recommended milk, but generally the general thing is that we should include at least 50% of the our plate should be of fruits and vegetables. Next slide. So this is an example of how the plate should be in real life. Here we can see that 50% of the plate is more of fruits and vegetables in both the plates, and it has one bowl. The first plate shows one bowl of dal, one bowl of sabji, some fruit like pomegranate and vegetables, and 1/4 is our whole grains. On the next plate, we can see again the same fruits and vegetables and some dal, like some pulses, source of protein, and whole grain, and some curry. So this is how we can actually include more of whole grains and to include more of the fiber in our diet. Yes, thank you so much. Sindhura ma'am, you can take away for the vote of thanks. Thank you so much.
Thank you so much, Deshan. Alright, so thank you so much everyone for taking your time out and attending this journal club. So I hope you found this session insightful. If you have any questions, you can please drop them in the chat box. We'd love to take it. And if possible, please try to share your suggestions, your feedbacks, your reviews in the chat box. That will be really helpful so that we can keep improving ourselves and keep coming with, even more better and better journal club in the further future journal clubs. Please do let us know how you felt about today's session. And if you have any questions, please feel free to drop it in the chat box, please. Can we have a recording of the session, please, for refreshing ourselves? Sure, Dr. Shobha. This will be uploaded in our YouTube channel. So once it's uploaded, we will let you know in the WhatsApp group. Yeah, hope that answers your query as well, Dr. Vasan. So we'll be sharing that. Once it's uploaded in the YouTube, we'll be sharing the link as well. So any inputs, feedback, so that we can improve ourselves, or any topics that you want us to present or take in our future journal clubs, please do mention that as well. We would love to take up those topics and present some very groundbreaking studies on the topics as well. Thank you so much, Dr. Vasan, Dr. Chandini. Thank you so much. Very good session. Keep going. These kind of sessions will help us. Thank you so much. We'll definitely do. Yeah, thank you. Thank you, Dr. Vindya. Thank you, Dr. Siddha. Thank you so much. Yeah. All right, if there are no more queries, so this is it for this month. So we'll be coming up with another interesting and a groundbreaking study in the next month. Please stay tuned. We'll be posting the updates. Thank you so much. Thank you so much, Dr. Manali. A most useful session. Thank you so much. Would like to know about autoimmune disorders in diet. Perfect. Noted, Dr. Chandini. So we'll definitely come back with one of the interesting studies on that topic as well. Thank you. So I'll be meeting. The GC team will be meeting you in the next month. Take care. Bye-bye. Good night.